In the spring of 2004, doctors at a hospital in Berlin were staring at test results they didn't believe. A 4-year-old boy sitting in the exam room had the muscle mass of a competitive teenage bodybuilder. His arms were carved with visible striations. His body fat was almost non-existent. He could hold a pair of 3-kilogram dumbbells out at arm's length, steady, for minutes at a time, something most grown adults in that hospital couldn't do. This wasn't a child who trained. He had never lifted a weight in his life. When geneticists sequenced his DNA, they found something that had only been theorized before that day. Both copies of a gene called myostatin, the gene responsible for putting the brakes on muscle growth, were broken. His body had no functioning brake pedal. It just kept building. His mother, a former professional sprinter, carried one broken copy herself, and it showed in her legs, unusually powerful, even by elite athletic standards. That one case, published in the New England Journal of Medicine, cracked open a question scientists had been circling for decades. If a single gene mutation could turn a toddler into a living strength machine, what happens when entire populations carry milder versions of that same genetic advantage, passed down for thousands of years? What if the reason certain nations produce sprinter after sprinter, or strongman after strongman, or marathoner after marathoner, isn't culture or coincidence at all, but something written directly into their DNA? That question is exactly what a growing body of genetic research has spent the last 20 years trying to answer. And what they found is going to change how you think about human performance forever. Stick around, because I dig into ancestry and DNA science every week. And this is one of the most fascinating and most argued about topics in the field. If you want more of this, hit subscribe, because we're about to get into gene by gene, population by population, exactly where these advantages come from and why some of them are almost unbelievable. Let's start with the basic biology, because you need this to understand everything else. Human skeletal muscle is built from 2 main types of fiber. Type 1 fibers, the slow twitch fibers, are endurance machines. They contract slowly, resist fatigue for hours, and are loaded with mitochondria, the cellular engines that burn oxygen to produce energy over long stretches of time. They're rich in a protein called myoglobin, which gives them a deep red color and lets them store oxygen for sustained use. These are the fibers that carry a marathon runner through mile 20 and a cyclist through a 6-hour mountain stage. Then you have type 2 fibers, the fast twitch fibers. These contract explosively, generate huge amounts of force almost instantly, and burn out fast because they rely on anaerobic metabolism instead of oxygen. They're larger, paler, and built for short, violent bursts of output. Within that category, there's a further split. Type 2A fibers are hybrids, able to switch between aerobic and anaerobic energy production, giving athletes moderate power that can be sustained a bit longer. Type 2X fibers are the purest power fibers in the human body, capable of maximum force output but fatiguing within seconds. Elite powerlifters and sprinters tend to carry unusually high concentrations of these. Here's the part that matters most. The ratio of type 1 to type 2 fibers in your body is set largely before you're born, and training cannot fundamentally rewrite it. You can improve the fibers you have. You cannot convert a body built for endurance into a body built for raw power, or vice versa, no matter how hard you train. That ratio is coded into you. And when researchers started sequencing muscle biopsies from thousands of athletes and comparing the genes tied to fiber composition, oxygen use, and recovery speed across populations grouped by ancestry, a pattern emerged that was too consistent to ignore. The first gene that comes up in almost every conversation about sprinting is ACTN3, sometimes nicknamed the speed gene. ACTN3 produces a protein called alpha-actinin 3, found specifically in fast twitch muscle fibers, where it helps them generate rapid, forceful contractions. Some people carry a variant of this gene, commonly called the X allele, that produces no functional protein at all. Others carry the R allele, which does. People with 2 copies of the R allele, known as the RR genotype, tend to have a real measurable advantage in explosive power-based movements, sprinting, jumping, and heavy lifting. Multiple studies going back to the early 2000s have found that elite sprinters and power athletes are disproportionately likely to carry at least one R allele, while endurance athletes are more likely to carry the X variant. And the frequency of that R allele isn't evenly spread across the globe. Research on West African populations has repeatedly found the R allele at very high frequency, and studies specifically on Jamaican sprinters have found it at levels among the highest recorded anywhere on Earth, even higher than in some other West African groups. Meanwhile, European populations sit somewhere in the middle, and some East Asian populations show the R allele at meaningfully lower frequencies. This doesn't mean everyone with the RR genotype becomes an Olympic sprinter, and it doesn't mean everyone without it can't be fast. But at a population level, the statistical tilt is real, replicated across independent labs, and it lines up with exactly the events those populations dominate on the track. It's not just about raw power output either. Populations with high frequencies of the ACTN3R allele also tend to show favorable variants in genes tied to inflammation control and recovery, like interleukin 6, a signaling protein that plays a dual role in the body. During exercise, it helps muscle cells pull in glucose and burn fat for fuel. After exercise, it kicks off the repair process and helps clear out inflammatory byproducts. Certain IL-6 variants, more common in West African and some Mediterranean populations, according to more recent research out of institutions studying elite athlete cohorts, appear to speed up that recovery process. Combine a genetic edge and explosive power with a genetic edge and bouncing back faster between training sessions, and you start to see how a small, specific set of gene variants can compound into a massive real-world advantage over a career, race after race, season after season. Now flip to the other side of the world and a completely different genetic story shows up. Northern Europe, especially Scandinavia, Iceland, and the Baltic states, has produced generation after generation of elite strongman competitors, powerlifters, and wrestlers. And when researchers looked at the genetics behind that, myostatin came up again and again. Remember, myostatin is the protein that acts as a brake on muscle growth. It stops your body from building unlimited muscle mass, which historically was useful because massive muscle is expensive to maintain in calories. But certain populations carry partial variants of the myostatin gene that dial that breakdown. Not off entirely, like the German toddler, but reduced. Studies of competitive strongman athletes in Iceland have found a notably high proportion, carrying at least one copy of a myostatin variant, associated with reduced myostatin activity and greater muscle mass potential. Combine that baseline genetic tendency with a cultural tradition that has produced world's strongest man champions out of a country with a population smaller than many mid-sized American cities, and you get a nation punching wildly above its weight in strength sports almost every year, for decades. Northern European populations also evolved something else that plays into this, tied to sunlight rather than muscle directly. In regions with long, dark winters and limited sun exposure for much of the year, populations evolved variants of the vitamin D receptor gene, VDR, that extract more benefit from the small amount of sunlight available and use it more efficiently. Vitamin D isn't just a bone health nutrient. It plays a direct role in muscle contraction, muscle repair, and testosterone regulation. Populations without efficient vitamin D processing living through 6-month winters would have faced real declines in strength and muscle function every year. The populations that adapted efficient VDR variants held onto more of their strength and hormonal function through the darkest months. That adaptation, layered on top of the myostatin story, helps explain why a disproportionate number of world-class strength athletes trace back to exactly these regions. East Asian populations tell yet another story, and it's not about raw power at all, it's about efficiency. Athletes from China, Japan, and Korea have historically dominated sports demanding precision and repeatable technical execution under sustained physical stress. Certain forms of gymnastics, table tennis, badminton, weightlifting technique categories, and various endurance precision hybrids. Genetically, researchers have pointed to variants of PPRGC1A, a gene that regulates mitochondrial biogenesis, essentially controlling how efficiently your cells produce and use energy. This gene doesn't make muscles bigger or faster. It makes the energy system running underneath those muscles more efficient, allowing sustained output with less waste and slower fatigue accumulation. It's a completely different kind of advantage from the ACTN3 story, and it shows up in completely different sports, in athletes who aren't necessarily explosive, but who can repeat the same demanding movement with precision thousands of times without breaking down. Researchers studying elite table tennis and badminton players in China have noted that this efficiency advantage compounds over a career in a way that's easy to overlook. An athlete whose muscles waste less energy per repetition can train longer, recover faster between sessions, and rack up dramatically more total practice repetitions across a decade than a competitor whose muscles are burning through energy stores faster for the same output. Over 10 or 15 years of a competitive career, that gap in cumulative training volume can matter just as much as any single explosive advantage. South Asian populations, from India, Pakistan, and Bangladesh, show a genetic profile tied less to explosive strength and more to functioning in extreme heat. Heat shock proteins, a family of proteins including HSP70, help cells survive and keep functioning under thermal stress by protecting other proteins from damage when body temperature rises. Elevated frequencies of favorable heat shock protein variants in South Asian populations correlate with better performance and endurance in high heat and humidity, conditions that would push most other populations into rapid fatigue and dehydration. It's a survival adaptation to a punishing climate that happens to translate directly into athletic resilience under heat. Polynesian populations, spanning Samoa, Tonga, and Hawaii, present something researchers still don't fully understand at the mechanistic level, but the pattern is unmistakable. These populations show some of the highest average bone density and lean muscle mass of any studied group on Earth, a trait that shows up clearly in the disproportionate number of elite rugby players, American football linemen, and strength-based athletes of Polynesian descent relative to the tiny population size of the Pacific Islands. Some researchers have pointed to variants in genes affecting bone growth and joint structure, alongside genes that historically promoted efficient fat and energy storage, an adaptation likely shaped by generations of long, resource-scarce ocean voyages where the ability to store energy efficiently meant the difference between survival and starvation. In a modern athletic context, that same energy storage efficiency shows up as a baseline advantage in size and physical mass. Look at professional American football today, and the pattern jumps out immediately. Players of Samoan descent alone make up a wildly disproportionate share of NFL rosters relative to how small the Samoan population actually is worldwide, a statistic that sports scientists have pointed to repeatedly when discussing how concentrated certain genetic profiles can become within a small, historically isolated population. Rugby tells the same story across Tonga, Fiji, and New Zealand, where Pacific Islander players routinely post some of the highest lean mass and bone density readings recorded in professional sport, again, without needing to explain it through training alone, since many of these athletes are compared directly against similarly trained peers of other ancestries and still come out ahead on raw physical measurements. Then there's altitude, and this is where the genetics get almost science fiction level. Indigenous populations of the high Andes in Bolivia and Peru live and function normally at elevations above 13,000 feet, where oxygen levels are roughly 40% lower than at sea level. Most people transported suddenly to that altitude would struggle to walk up a flight of stairs. Andean populations have adapted over thousands of years with elevated hemoglobin concentrations that let their blood carry more oxygen per volume, along with cardiovascular adaptations that keep their hearts and lungs working efficiently despite the thin air. Interestingly, high-altitude populations on the other side of the world, in Tibet, evolved a different genetic solution to the exact same problem. Tibetans carry a variant of a gene called EPAS1, which regulates the body's response to low oxygen, and rather than dramatically increasing hemoglobin the way Andean populations do, the Tibetan variant lets their bodies function efficiently at altitude without the blood-thickening side effects that usually come with it. Genomic research has traced part of that Tibetan EPAS1 variant back to interbreeding with Denisovans, an extinct human relative, tens of thousands of years ago, meaning some of the most useful high-altitude genetics on the planet may have entered the human gene pool through a completely different species. Two populations, facing the identical survival problem of extreme altitude, solved it through entirely different genetic pathways. That's not culture. That's evolution running two separate experiments and landing on two separate answers. What makes this even stranger is how recently we discovered it. For most of the 20th century, physiologists assumed high-altitude adaptation worked the same way everywhere, through simply pumping out more red blood cells. It wasn't until genomic sequencing became affordable in the 2010s that researchers realized Tibetan populations had essentially found a shortcut, achieving the same functional oxygen delivery without paying the cardiovascular cost that comes with permanently thickened blood. Andean populations, despite living at comparably brutal elevations for a comparably long stretch of human history, never evolved that same shortcut, and instead carry a higher lifetime risk of chronic mountain sickness as a trade-off for their hemoglobin-heavy strategy. Same mountain, same oxygen problem, two different evolutionary bills to pay. Middle Eastern populations across the Levant, the Arabian Peninsula, and North Africa show genetic adaptations centered on heat regulation and cardiovascular efficiency in extreme dry heat. Variants in genes like ACE, which affects blood pressure regulation, have been studied in relation to performance under thermal stress, and Bedouin populations in particular have been documented sustaining physical activity in desert conditions that would incapacitate most outsiders within hours through more efficient water conservation and more stable core temperature regulation during exertion. There's also a sex-based layer to this story that researchers have only started untangling more recently. Within any given population, men and women show different baseline hormonal environments, particularly testosterone, that interact with these same genes in different ways. A woman carrying the ACTN3RR genotype and a man carrying the identical genotype won't necessarily express the same magnitude of power output because testosterone influences muscle fiber size and neuromuscular recruitment independently of which gene variants you're carrying. That doesn't cancel out the population-level patterns researchers have found, but it does mean the full picture requires looking at genetics, hormones, and training history together rather than treating any single gene as destiny on its own. It's one more reminder that even the most striking gene, taken in isolation, only ever tells part of the story. Now here's the part of the story that makes it genuinely controversial, and I want to be straight with you about why. Whenever genetic research groups results by ancestry or population and finds real, measurable differences, it walks straight into one of the most sensitive debates in all of science. Critics have argued, for good reason grounded in a dark history, that highlighting population-level genetic differences can be twisted to justify discrimination or revive discredited ideas about racial hierarchy from a century ago. That history is real, and it's why researchers in this space have to be extremely careful about how findings get framed and communicated. But the underlying genetics themselves, the ACTN3 frequency differences, the myostatin variants, the EPAS1 story in Tibet, the heat shock protein patterns in South Asia, these are not fringe theories. They've been published in peer-reviewed journals, replicated across independent research groups, and they don't support a story about any population being globally superior. What they support is a much more interesting and much less politically loaded idea. Different environments, over tens of thousands of years, shape different bodies in different directions. There is no single best genetic profile for human performance. There are dozens of profiles, each optimized for a different environmental pressure, a different climate, a different survival challenge, and by extension, a different athletic event. And genetics is only half the story anyway. Environment and culture do the rest of the heavy lifting, and nowhere is that clearer than in Jamaica. Jamaica has a population of under 3 million people, smaller than many single American cities, and yet it has produced more world-class sprinters per capita than any nation on the planet. Yes, the ACTN3R allele frequency in Jamaican populations is extraordinarily high, among the highest ever recorded, and some researchers have theorized that population bottlenecks during the transatlantic slave trade and the generations that followed may have concentrated that genetic profile even further. But genetics alone doesn't explain what happens on that island. Jamaica has built an entire cultural infrastructure around track and field. The high school championships, known nationally as champs, draw crowds well over 30,000 people and get treated like a professional sporting event, broadcast nationally, scouted by international coaches, covered obsessively in local media. Teenage sprinters become national celebrities before they finish high school. Every parish has a track club. Every serious young runner has a visible, well-worn pathway from local meets to champs, to national teams, to the Olympic stage. The genetic raw material exists elsewhere in West Africa, too. But Jamaica is the place that turned it into a national identity. And that combination of biology and culture is what actually produces the sprinting dynasty the world watches every 4 years. Kenya offers an almost identical lesson from a completely different genetic starting point. Within Kenya, one ethnic subgroup, the Kalenjin, makes up a relatively small share of the national population, but accounts for a wildly disproportionate share of the country's world-class distance runners. Anthropological research on Kalenjin runners has documented distinctly long Achilles tendons and thinner lower legs, a body structure that reduces the energy cost of each stride over long distances, along with a higher proportion of slow-twitch, endurance-oriented muscle fiber. But layered on top of that biology is an environment built almost perfectly to develop it. Many Kalenjin children grow up at high altitude in the Rift Valley, running long distances to and from school every single day, in a culture where distance running is both deeply respected and, for many families, one of the clearest paths to real economic opportunity. Strip away either half of that equation, the genetics or the environment, and the story falls apart. It's both, working together, that produces the outcome. This is where the science is heading next, into a field called epigenetics, the study of how environmental factors can switch genes on or off without changing the underlying DNA sequence itself. Training, diet, altitude exposure, even chronic stress can all trigger epigenetic changes that either amplify or suppress a genetic advantage a person was born with. A child born with elite sprinting genetics might never fully express that potential if they grow up somewhere with zero coaching infrastructure and no cultural pathway into the sport. Meanwhile, someone with a more average genetic starting point can, through smart training and the right environment, push performance further than raw genetics alone would predict. The DNA sets the ceiling. Life decides how close you get to it. And there's a much thornier question sitting right behind all of this. If scientists can now identify many of the specific gene variants tied to explosive power, endurance efficiency, or oxygen use at altitude, gene editing technology is advancing fast enough that some researchers are openly asking whether those genes could eventually be edited directly in embryos or even in already born athletes. CRISPR gene editing tools have become dramatically cheaper and more precise over the past decade, and the same techniques being explored for treating genetic diseases could, in theory, be pointed at genes like myostatin or ACTN3. That's not science fiction anymore. It's a live ethical debate happening right now in genetics and sports medicine circles, and it raises uncomfortable questions nobody has clean answers for. If genetically engineered muscle composition becomes possible, does that break competitive sport as we know it? Would we need entirely separate categories for edited and non-edited athletes? And if certain populations already carry measurable natural genetic advantages before anyone touches a gene editor, is elite sport already less level than we like to pretend it is? Some researchers have floated timelines suggesting the first generation of embryos selected specifically for athletic gene variants could exist within 20 years, not through direct editing, but through embryo screening technology that already exists for disease prevention and could, in theory, be repurposed to flag embryos carrying favorable copies of ACTN3 or myostatin. Sporting bodies like the World Anti-Doping Agency have already begun discussing what a genetic testing framework for competition might even look like, years before the technology is fully mature, precisely because they don't want to be caught unprepared the way they were with early generation doping decades ago. Sports have always celebrated natural biological advantages without much controversy. Nobody argues it's unfair that Dutch and Dinaric basketball players tend to be exceptionally tall, or that certain swimmers are built with unusually long torsos and wide wingspans, or that elite gymnasts tend to have shorter limbs relative to their torso, giving them better rotational control. Height, limb proportion, wingspan, these are all genetic gifts that sport simply accepts as part of the game. The conversation only gets uncomfortable when that same kind of biological variation lines up with ancestry instead of individual body type, because ancestry carries centuries of political and historical weight that height in basketball simply doesn't. But the discomfort of the conversation doesn't make the underlying biology less real. Human populations spent tens of thousands of years spread across radically different environments, facing radically different survival pressures: altitude, heat, cold, famine, disease. Evolution responded to every single one of those pressures differently in different places, and some of those responses happened to translate, thousands of years later, directly into athletic performance. None of this means any population is superior to another. It means the opposite, actually. It means human genetic diversity is real, deep, and remarkably specific, and every population on Earth carries a different set of adaptations forged by a different environment. The Kalenjin runner's tendons, the Icelandic strongman's myostatin variant, the Tibetan climber's oxygen efficiency, the Bedouin's heat tolerance, the Jamaican sprinter's fast twitch dominance, none of these cancel each other out or rank against each other. They're different answers to different questions the planet asked different groups of people over tens of thousands of years. That diversity isn't a flaw in the human genome. It's the clearest evidence we have of how adaptable our species really is, and it's the same diversity that gives humanity resilience against disease, the ability to survive in nearly every climate on Earth, and raw material for whatever challenges come next. Your DNA didn't get to choose your ancestors' environment, but it inherited the solutions they evolved to survive it. Understanding those solutions doesn't diminish anyone. It just tells a more accurate, more fascinating story about where human performance actually comes from, and how much more we still have to learn about the code written into every single one of us. Every year, new sequencing studies add another layer to this picture, and every year, the story gets a little more complicated and a little more humbling. There is no final answer waiting at the end of this research, no single gene that explains greatness, and no population that holds a monopoly on it. What there is, instead, is a species that spread itself across every corner of a difficult planet and came back, again and again, with a different biological solution to survive it. Athletic dominance, in the end, is just one small, visible echo of that much larger story.